Welcome to the second session on vaccines for TB. In the first session, you must have heard about the limited efficacy of the BCG vaccine, and all of you will agree that there is a need for newer TB vaccines. Why? Because the WHO Sustainable Development Goals and NTB strategy recommends the vision as a world of zero TB deaths, disease and suffering due to TB. And we are targeting about 95% reduction in the number of TB deaths, that is TB mortality, and 90% reduction the incidence of TB by 2035 as compared to the 2015 figures. So, in the slide on epidemiology of TB and in that talk, you would have heard that the current incidence of TB globally is about 10 million, and we need to reduce that incidence of TB. So, modeling studies have shown that to reach that to 2035 target of TB reduction, we have to do a lot of things. So, the first thing is, if you look at the y-axis, you can see the TB rate per lakh per year, and this blue line down is the line where we reach the targets of 2035. So, currently the incidence of TB is beyond 100 per lakh per year, and if we continue with the same trend, then currently the decrease is about 1.5% per year. If we continue with this trend of decline, then at 2035 we will be nowhere reaching this target. However, at 2020, introducing new tools emerging from the pipeline, optimizing the use of current tools, pursuing universal health coverage and social protection would reduce the incidence to minus 10% per year by 2025. So, if you would leave it like minus 10% per year, then it would go and follow this line wherein the decline would be minus 5% per year. Still we would have not reached the 2035 targets. So, at 2025, we need to introduce new tools, vaccine, new drugs, treatment regimens for active TB disease and latent TB infection along with the point of care test. So, this is known as the bending of the curve, and when we introduce all this by 2025, then you can see there would be a drastic decline of minus 17% of TB incidence per year, and wherein by 2035 we would reach the targets for the NTB strategy. So, in this context, introducing new tools, vaccines is a priority for reducing the burden of TB. So, what is the attribute of an ideal TB vaccine? So, this vaccine that we are going to develop or use it should be safe and efficacious at high risk infants, children and adults. So, by efficacious we mean that it should effectively prevent the disease or infection or recurrence or many other aspects for which it is developed. And it should be effective against all forms of TB like pulmonary, extrapulmonary TB, drug sensitive and drug resistant TB. Moreover, it should be logistically feasible to administer this vaccine under field conditions. So, a temperature that it could be kept in the normal fridge that can be used under the field settings would be an ideal requirement for a vaccine for it to be implemented widely. And the formulation must be quite easy so that it can be manufactured at a larger scale. We would require large quantities of the vaccine because India is a country that is accounting for 25% of the global TB burden, and so we need to give it for a larger at-risk population. And the storage and administration and it should have only low technology conditions for storage and administration. So it is practically feasible under the field settings. So, what are the different strategies for TB vaccination? So, we have pre-exposure strategy and post-exposure strategy. So, what do we mean by pre-exposure and post-exposure? So, the population for pre-exposure is MTB uninfected. So, MTB uninfected implies that this population is free of TB infection. So, infection can be measured by the tuberculin skin test or the IGRA test, that is interferon gamma release assay, and that you would have heard it in a different session about the two different tests. So, in this population, the vaccine mainly is to develop to prevent infection and prevention of disease. So, POI refers to prevention of infection and prevention of disease, and the population is the uninfected population. In terms of post-exposure strategy for TB prevention, first we have the population that is infected. So, in the target population of infected, that is those who are TST positive or IGRA positive, so the vaccine's main aim would be to prevent disease occurring in them, that is POD, prevention of disease. Subsequently, we have a group, a target population with TB disease, and we have therapeutic vaccines in them. So, what does this therapeutic vaccines do? They are aimed to improve treatment outcomes, and they are used as adjuncts along with TB treatment, etc. And the last group is those who are treated for TB. So, in this people, the aim of the vaccine should be prevention of TB recurrence, that is POR. So, POI is prevention of infection, POD is prevention of disease, and POR is prevention of recurrence, wherein what do we mean by recurrence is once a patient getting cured and not getting the disease back. So, it should prevent the person from getting the disease back, that is the prevention of recurrence. So, for each intention for which the vaccine is developed, the target population would be different, and as we know, the risk of TB infection and disease increases with age. One another point to be made is a therapeutic vaccine can also act to prevent the recurrence also. We hope we are clear for different strategies for TB vaccination. Now going on to the different type of TB vaccines. So, there are different ways vaccines are developed and for better efficacy, and we have the TB subunit vaccine, which is here. And what do we mean by subunit vaccine? So, these vaccines have immunoactive ingredients, and these include proteins that are isolated from and purified from Mycobacterium tuberculosis, and the property of this vaccine is enhanced by using adjuvants. So, the different vaccines that are developed using this platform are M72 AS01, GAM-TB-VAC, H56IC1 IC31, H4IC31, and ID93 plus GLA-SE. So, these are all subunit vaccines. And the next we have the recombinant live vaccine. So, the recombinant live vaccine uses a live vector to deliver the heterologous antigens. And so, this viral vector, it acts as a vehicle or a delivery unit for this antigen, and immune responses are elicited towards this antigen being presented, and also because of this viral vector. So, these are all called recombinant live vaccines, and we have different vaccines developed based on this platform, that is a TB FLU01L, FLU04L, AD5AG 85A, CHADOX1 85A, MVA 85A. Next, we have the attenuated live vaccine. So, what do we mean by attenuation? Is actually the weakened version of the living organism. So, the living organism, that is the bacteria, is weakened so that it is not causing disease when it is administered. So, that is attenuated live vaccine, and that is MTB-Vac now. The next is inactivated TB vaccine. Under inactivated TB vaccine, either the whole bacteria or certain fragments are prepared physically and chemically, and they are tested for immune responses. So, under that we have DAR 901, RUTI and VACI. The next is the DNA vaccines, and these consist of a plasmid, a promoter, multiple cloning sites and antibiotic. We will not go much in detail, but this is another platform wherein vaccines are also developed and has the advantage of wide variety of routes by which it can be tested, like mucosal, intramuscular, intradermal, transdermal, etc. So, these are the different vaccine platforms by which the vaccines are being developed. Now, when we come to the TB vaccine pipeline, so we have a lot of vaccine candidates that are either in the preclinical, phase 1, phase 2 and phase 3. So, if we take clinical trials, preclinical studies are actually done in animals. So, preclinical means before coming to humans. So, once if it comes to clinical trials, then it starts with phase 1, and this is basically in human participants. So, in terms of preclinical, you can see there are a lot of vaccine candidates. Phase 1 studies are usually done in healthy volunteers, and this is mainly focused on safety of the vaccines, looking at what particular dose the vaccine is safe, etc. The next is phase 2 study. So, phase 2 studies basically look at the immunogenicity as well as safety of the vaccine. The immunogenicity, by immunogenicity we mean both the humoral and cellular antibody responses have to be being tested in this phase 2 studies. And in phase 3 studies, what happened is the target population is involved in this studies. Both phase 2 and phase 3, the target population is involved, that is the population in which the vaccine is intended to be used. In phase 2, it is more of immunogenicity and safety, whereas in phase 3, one looks at whether the vaccine is efficacious against prevention of disease or prevention of infection, or for what it is developed for, as I had mentioned earlier during the strategies for vaccine development. Now, in the subsequent few slides, we can see the vaccines and the different phases of clinical trials in which they are. So, MIP, that is a Mycobacterium indicus prani, this is a whole cell Mycobacterium indicus prani heat killed vaccine. So, right now MIP and VPM, so these two vaccines are tested against the placebo in the prevention of disease, that is, this is a POD vaccine among household contacts who are aged more than or equal to 6 years, HIV negative, and this trial is currently ongoing in India and has a population of over 12,000 household contacts. And the next is the VPM 1002, it is a live recombinant BCG vaccine where the urease C gene is replaced by listeriolysin O gene, O encoding gene, that is HLY, from the Listeria monocytogenes. So, this vaccine is tested along with MIP in the previous trial which I had mentioned. And whereas in other countries like South Africa, this is being evaluated as a prevention of infection vaccine against BCG in preventing MTB infection among newborn infants who are HIV exposed and uninfected. So, more than 6,000 infants are being studied in this trial. And there is another study in India and Bangladesh in which the prevention of recurrence, wherein in about 2000 TB patients aged 18 to 65 years who have been treated, this vaccine is given to see if it can prevent the recurrence among them. The next we go to the M72 ASO1E vaccine, which is a protein adjuvant subunit vaccine. And currently a phase 3 trial, a large scale phase 3 trial is planned in 20,000 individuals aged 16 to 34 years with and without MTB infection with the strategy of prevention of disease. The MTB vac is another vaccine which is live and genetically attenuated MTB, and this is compared with BCG in around more than 6,000 HIV unexposed and exposed infants in South Africa, Senegal and Madagascar in the prevention of disease. BCG revaccination. So, primary BCG vaccination we saw had very limited efficacy, and currently the interest is BCG revaccination and to see if it can prevent infection, and the study is going on in phase 2 in South Africa in prevention of infection in BCG vaccinated MTB uninfected adolescents. As a pre-travel vaccination among 2000 BCG naive MTB uninfected adults, either healthcare workers or long-term travelers, in the strategy for prevention of infection. The GAM-TB vac is a protein adjuvant subunit vaccine, and it is tested against the placebo in preventing primary TB disease among more than 7,000 HIV negative BCG vaccinated MTB uninfected adults in the Russian Federation for prevention of disease. So, those are the phase 3 trials. So, the main thing is to prevent disease or prevent infection, which is the main objective of those trials. Now, phase 2 trials. So, in phase 2 trials, as I said earlier, this looks mostly at looking at immunogenicity as well as safety, and to an extent prevention of infection or disease or recurrence, and for which it is developed for. So, DAR901 is an inactivated whole cell vaccine, and this study has been completed. It was tested against placebo in the prevention of MTB infection in 650 BCG vaccinated HIV negative MTB uninfected adolescents. And H56IC31, this is a protein subunit vaccine. It is being evaluated for prevention of recurrence in HIV negative adults who have completed at least 5 months of drug susceptible treatment in South Africa. So, the ID93 GLA-SE vaccine, which is sponsored by Curatis, which is a protein adjuvant subunit vaccine. This has completed a phase 2 safety, immunogenicity, efficacy study comparing the low and the high dose with the placebo in 107 BCG vaccinated MTB uninfected healthcare workers in South Korea. The same vaccine sponsored by NIH is there is a plan for a phase 2A, 2B immunogenicity study as a therapeutic adjunct in 1,500 HIV positive negative people being treated for drug sensitive TB as a prevention of recurrence vaccine. The RUTI vaccine, which is a fragmented MTB, this is being tested against placebo as a therapeutic adjunct to 140 HIV negative adults undergoing treatment for drug susceptible TB and MTB in India. The CHADOX185A plus MVA85A vaccine, which is a viral vector vaccine, this is undergoing a phase 1-2 dose escalation and age de-escalation safety study in adults and adolescents in Uganda. So, coming to phase 1 trials, so this mainly looks at safety of the vaccine and mainly done in healthy volunteers. So, we have the BCG aerosol. This has completed a phase 1 study at different doses delivered via aerosol or intradermal route versus placebo in 46 BCG naive adult volunteers in the United Kingdom. We have the viral vector vaccine, which is ADHU5AG85A, which is being tried in the aerosol route, and it has completed the phase 1 study of high and low dose in 36 BCG vaccinated adult volunteers in Canada. The AEC-BC02, which is a protein adjuvant subunit vaccine, is undergoing a phase 1B study looking at the low dose as well as the adjuvant versus placebo in 30 adult volunteers in China. The TB flu vaccine, which is a viral vector vaccine, has completed a phase 1 study versus placebo in 44 BCG vaccinated adults and a phase 1 study in the intranasal and sublingual administration among the 36 BCG vaccinated adults. So, we can see not only intradermal route, but other routes like aerosol, sublingual, intranasal, different routes are also being studied for vaccination for TB. So, we have a lot of challenges in the development of new vaccine because there is a large amount of gaps in knowledge of the immune system in TB. We do not have good correlates for protection like a good biomarker which can say this vaccine is really good, and there is no consensus on suitable animal model as far as TB research for drugs or vaccine is concerned. And another issue is the availability of funding. So, right now there is a lot of efforts being put into research and development considering the timelines for TB elimination, but the funding is still not optimal, and many TB endemic countries lack infrastructure for large clinical trials. So, as you see, the vaccine has to undergo rigorous testing in clinical trials, and the capacity for clinical trials has to be built, and this has to be in place for new TB vaccine development. So, coming to the key vaccination, in order to achieve the end TB strategy, prevention through vaccination is the most cost effective tool for TB elimination and controlling TB. And as we saw in the earlier session, BCG is a currently available vaccine for TB which offers modest protection for disseminated TB in children. New TB vaccines is the single greatest preventive tool, and without them TB cannot be eradicated. And as we saw in this session, some promising vaccine candidates are being studied in clinical trials. So, I hope that this session is useful, and India has a target for TB elimination in 2025, and as we saw earlier, for bending the curve and achieving the targets for TB elimination, it is very important that new vaccines are rolled out and the burden of TB is reduced in our country. Thank you very much for your attention.